Imagine waking up tomorrow and learning that scientists have found a way to rewind the biological clock inside every one of your cells without touching your DNA. Sounds like science fiction, right? Yet a quiet revolution is unfolding at Boston Children's Hospital, and it centers on the tiny shoelace-like tips of your chromosomes called telomeres. These microscopic aglets protect your genetic material the same way plastic tips keep shoelaces from unraveling. And when they wear down, the entire fabric of your body begins to fray. But here's where it gets controversial. Some researchers now believe we can patch those tips back on once and for all. What exactly are telomeres, and why should you care? Think of each chromosome as a long, delicate thread. Every time a cell divides, a few millimeters of that thread are snipped off the end. Telomeres are the expendable caps that take the hit, so the vital genes farther down the thread stay intact. Over decades of cell division, the caps shorten until they can no longer protect the chromosome. At that point, the cell either retires, a state called senescence, or self-destructs. In healthy aging, this gradual loss is normal. In people with telomere biology disorders, TBDs, such as the rare but devastating dyskeratosis congenita, the caps disappear at breakneck speed, leading to bone marrow failure, lung scarring, or liver disease while patients are still in their 20s or 30s, the enzyme that could change everything. For more than 10 years, Dr. Suneet Agarwal, a hematologist and stem cell researcher at Boston Children's, has chased a single question. Can we persuade the body's own maintenance crew to rebuild those caps? The crew in question is an enzyme named telomerase. Under normal circumstances, telomerase is switched off in most adult cells, which is one reason telomeres erode over time. Reactivating telomerase sounds simple, until you realize its RNA blueprint, called TERC, is enormous, twisty, and notoriously fragile. Enter Neha Nagpal, PhD, a chemical biologist who specializes in taming unruly molecules. Nagpal and Agarwal devised an enzymatic trick that staples TERC into a sturdier, synthetic version they call engineered TERC, or eTERC. When they slipped this reinforced blueprint into human stem cells, something remarkable happened. After a single treatment, telomeres lengthened and stayed longer for roughly 69 days, biological dog years compared with the human lifespan. Even more surprising, the cell's other housekeeping systems hummed along undisturbed. No runaway growth, no genetic chaos, just a temporary targeted tune-up. But here's the part most people miss. eTERC doesn't stick around forever. It delivers its instructions, boosts telomere length, and then politely exits the stage. Agarwal likes to call it a one-and-done intervention, a phrase that makes some longevity enthusiasts cheer and some ethicists raise an eyebrow. After all, if we can top up telomeres at will, where do we draw the line between therapy and enhancement? From lab bench to bedside, the delivery dilemma. The next hurdle is sneaking eTERC into living tissues. RNA is notoriously picky about where it will and won't go. Agarwal's team is experimenting with nano-sized lipid bubbles and small molecule escorts that can shepherd eTERC across cell membranes without triggering immune alarms. If they succeed, the same approach could one day treat not only TBDs but also common age-related conditions tied to short telomeres. Think heart disease or early dementia. Skeptics argue that artificially lengthening telomeres might increase cancer risk since many tumors rely on telomerase to become immortal. Agarwal counters that eTERC's transient nature and built-in safety switches should prevent such runaway growth, but only large-scale trials will settle the debate. A second path: feeding the repair crew. While eTERC works at the blueprint level, another strategy simply hands the repair crew more bricks and mortar. Agarwal's earlier studies showed that flooding cells with two common nucleosides, thymidine and deoxycytidine, can dramatically elongate telomeres in lab dishes and in patient-derived stem cells. The concept is elegantly low-tech. Give cells the raw ingredients they need to rebuild their caps. Now, a first-in-human Phase 1 trial is poised to test whether a thymidine plus deoxycytidine cocktail is safe for children and adults with TBDs. If the therapy proves both safe and effective, it could become the first FDA-approved treatment that directly targets telomere length. Genetics meets reality. Why one size never fits all. Yet even the most ingenious therapy will face a moving target. TBDs are not caused by a single villain gene, but by dozens of rare mutations sprinkled across the genome. Two siblings can carry the exact same mutation and still follow wildly different clinical paths. One may need a bone marrow transplant in childhood, while the other lives symptom-free into old age. Why? Vijay Sankaran and graduate student Michael Poshla think part of the answer lies in the genetic background noise we all carry. Using data from the UK Biobank, they calculated polygenic scores. Think of them as composite credit ratings for telomere length, based on thousands of common genetic variants. They discovered that people with severe early-onset TBD often have not only the rare high-impact mutation, but also an unlucky collection of everyday variants that naturally shorten telomeres. In other words, your genetic lottery ticket isn't just the rare mutation, it's also the small print terms and conditions that come with the rest of your genome. This finding flips the traditional narrative. Instead of blaming everything on a single rogue gene, we may need to treat TBDs as complex, multifactorial diseases. The upside? Polygenic scores could one day help doctors predict which family members are most at risk and tailor monitoring or early interventions accordingly. The downside? Such scores might also raise thorny questions about insurance discrimination or reproductive decisions. So, what does the future hold? Boston Children's researchers are pursuing a three-pronged attack. 1. Transient telomerase boosters like eTERC. 2. Nutrient-based nucleoside therapies. And 3. Precision genetic risk profiling. Each path has champions and critics. Some bioethicists worry that even well-intentioned telomere extension could widen social inequalities if only the wealthy can afford it. Others fear a slippery slope toward designer babies with unnaturally long lifespans. Meanwhile, patients and families living with TBDs simply want more birthdays, more graduations, more ordinary moments that most people take for granted. Here's the provocative question I'll leave you with. If science hands us the tools to safely rewind our cellular clocks, should we use them only for disease, or also for the human desire to live longer, healthier lives? Is extending telomeres a moral obligation to alleviate suffering, or a Pandora's box we should keep firmly shut? I'd love to hear where you stand. Drop your thoughts below. Agree, disagree, or bring an angle no one's considered yet. After all, the next breakthrough might come not from a lab, but from a conversation like this one.